Okay. All right. So today, as a group for Adaptive Microbe, our group will be Group 12. My name is Asil Hisham from UPM 3. My metric number is 186446. So our group decided to choose Clostridium perfringens. For the reasons of our choice of this microorganism is because it's unique, yet it's quite common as a disease, especially here, especially for diabetic patients. There's something special about this microorganism. It is that it causes gas gangrene, which is when the human skin or animal skin, it happens in both, starts killing it, dying by itself, and causing acidic gas to be produced, as well as damaging the human tissues. It has a lot of factors. It causes a lot of problems, and this will be each person will share about any information they've gathered about this microorganism. So, who wants to start first?
My name is Farhana Saini. My 190189. I'm from UPM3.
Okay. Okay, Fatin,
Yes.
start talking.
About the microbe.
Yes, about the microbe, the Clostridium.
All I know about Clostridium is the major factor of food poisoning.
Oh yeah, it actually is. It causes food poisoning most commonly for internal issues. For external, it's more into any infection that occurs into the skin untreated. It could be, if it's infected by Clostridium perfringens and it's not treated immediately, it will cause gas gangrene. And a person infected with gas gangrene, they will have to amputate that limb. And when it causes food poisoning, the person has to be hospitalized for treatment. Okay. Anyone else? Hidayah? Oh, what's your opinion about this microorganism?
Yeah, this single cell organism is found pretty much everywhere. And yeah, I know it's a leading cause of food poisoning. So from my search in Google, they said that this organism is the fastest known for its reproduction rate.
Interesting. So this is very fast. Okay.
Oh, can you hear me?
Yes. Wonderful. Okay. Do you want to add anything else?
No, nothing.
Okay. Who else has points about this microorganism?
Me, Yuva, Asil, can you hear me?
Yes, Yuva, we can hear you. Okay, what do you think about this microorganism?
I want to know about the diagnosis method.
Okay.
Do you know how the diagnosis method works?
I think it's just
For this,
using something.
For this bacteria.
Okay. So what is it? I'm sorry, just Hanis, no wait. Someone's microphone is quite loud. Can you mute? Yeah, can you mute it so Yuval can speak louder? Okay. So Yuval, what's the diagnosis?
Yeah, I want to know about the diagnosis of this microbe. How to diagnose?
Okay. For the diagnosis, it's usually there's different things to diagnose this one. So first of all, it's usually the gram stain, which you have to do all the time, which is gram positive. But the perfect way to ideate is using Nagler's reaction. Well, this is something we need to do more research on. That is specifically used for the Clostridium perfringens, detection for diagnosis. So if the patient has that, so this body sample, either body fluid if it's by food poisoning, or pieces of tissue if it's gas gangrene, then it's cultured, then that sample is used for Naegler's reaction as the last part, because of course you need to do the normal culture, which is in anaerobic blood agar. So when you see double zone hemolysis, then after that you follow by motility. Motility test is motile, and the gram staining, gram positive bacilli, spore forming and capsulated. As well, then the final thing is identification by Naegler's reaction.
Thank you.
Yeah, okay. So anybody who has other points to add? Hanis?
Okay, I wanna ask who wanna do the toxic part. Actually, what's about the toxic? I'm not. I can't understand why we have to do what we have to do in the toxic part.
Okay, let's first share all the information that we have, and then we take part by part all together and discuss it. Okay. All right. Wait, Ashwin wants to speak. Yes, Ashwin?
Okay, my name is Ashwini. I'm from UM2, and my matrix number is SEQ150003. Okay, just to clarify on the toxins, this for Clostridium perfringens, there are four toxins. Yeah. So you have alpha toxins, beta toxins, epsilon toxins, and iota toxins. So each toxins, they are produced by different strains. So they have an 8 classification of strains. So,
Ashwin, can you just hold on for a second? Yeah. Can you check just your connection, because the voice is just cutting a little bit.
Sorry?
Your connection is cutting a little bit.
Oh, I'm sorry. I'm using data, so.
Oh, it's okay. Just keep the mic near your mouth and speak. Okay.
So now it's okay. Now it's better. Alright. Okay. So, alright. So, like I said, there's 4 toxins. So, like type A bacteria, type A strain bacteria would produce alpha toxins. Type B bacteria produce alpha beta toxins. Type C bacteria produce alpha and beta toxins, and type D bacteria would produce alpha and epsilon toxins, while type E bacteria will produce alpha and iota toxins. So each toxins, they have like different functions. They affect the bacteria and the host. So we can see more about that mind map, of course. Yeah, but that's basically what the toxins are about, just to clarify Yuva's question just now. Yeah.
Thank you, Annie.
Yeah, thank you. Okay. Hanis, would you like to add?
Yes.
Okay. Name and
Loud noise.
It's okay.
Okay. I found that even though the close.
Introduce yourself. Introduce yourself.
My name is Nurhanis. I'm from UPM2. My matrix number 189159. Okay, I found that even though the Clostridium can cause food poisoning, but it can be ingested and cause no harmful.
Okay. Okay, that's wonderful. Okay. Do you want to add something else?
No.
Anisha, you want to add something else?
My name Anissa binti Akbar from UPM2. My matric number 188821. So I'm gonna read about this bacterial morphology. It's about its cell wall. The bacteria that give a positive result in gram staining, gram-positive bacteria take up the crystal violet stain used in the test and then appear to be purple colored then seen through a microscope. This is because the thick peptidoglycan layer in the bacterial cell wall retains the stain after it is washed away from the rest of the sample in
Decolorization.
stage of the test. Thank you.
Thank you. So anybody else want to add anything about this microorganism?
Asil, I want to introduce myself. I forgot to introduce myself.
Oh, okay. Introduce yourself. Oh, sorry. Oh, I think we just got too into discussing about this microorganism. Okay, introduce yourself, please, and university and group.
Yuvarani Murugan from UKM. Metric number is E158763.
Okay. We have in our discussion group only 7. The other 2 members were not able to join because they have connection problems. That's why universities should add proper internet connection. So so far this is I think the basics of our very short-term research about Clostridium perfringens. So I think we need 2 days to do full research, and so we'll be capable of doing a mind map in which each person will choose a specific section to discuss. For example, there's a lot of sections to cover. Like, for example, the species classification, introducing from the kingdom until it's Clostridium Clostridium, yes. And then another person maybe will speak about the different locations where it can be found. So here we discovered that food poisoning and gas gangrene in body. So we can say we can find it in the intestinal or maybe the stomach. Second one is into open wound. So that's part of the location as well where this infection occurs. So maybe this infection occurs in maybe food beside water bodies or near hospitals. You following?
Yes.
Yeah, okay. So that's the division. Maybe the what effect it has on humans, what effect it has on animals, because it affects actually both humans and animals. So the different effects it has, and how do we detect it, which is using lab. So there's multiple tests we have to go through. So including the gram staining, the spore motility, normal anaerobic blood agar,
So
chocolate
basically,
agar, technique, and why is it so special? So what is something specific special about it? For example, if it's ingested, it does not cause harm as Hani stated, but when it causes tissue infection, if it's not cleaned properly or immediately, it causes amputation. So you can see it's like something completely normal, but at the same time it's very, very harmful. So depending on the location. So the 2 things are connected. And maybe we can find out the history a little bit, when it was first detected, and then what kind of diseases and symptoms. So that's also, and prevention. Is anything else should we cover as well?
Azil, I want to ask. I'm doing microscopy view. Then just I need to give the picture, or I need to give some procedure of the experiment, or how?
Oh, whatever. I think if you're doing it a mind map, I think you just need to add a picture and describe it. What do you guys think?
Yeah, that's
About
what
what?
I think
Because
too.
you guys already tell about it.
Oh, yeah, because we're sharing information. So maybe when you want to do the mind map, I'm thinking of, since the mind map is online, so each of us has the link. So each person adds their part and then maybe help the others. Maybe they see a part, for example, it can become better or change it. So we edit each other's work. So that way, each of us will learn from the others. At the same time, we help each other to get this done over.
Okay.
Okay.
Or maybe if like genome, not genome, sorry, you can always talk about the metabolism of
The metabolism and all that kind of things. Yeah, so we can divide these things for maybe, for example, like microscopy. You can see what maybe under light microscopy how it looks like, under electric microscopy how it looks like. something like that. So what else do you guys want to add?
The staining technique.
Yeah, true. Staining. We forgot about it because it's not only gram staining. So there's possibility there's other stains.
Yeah. Yeah, sure.
Yeah. So I think we need to do more research about this. So what else? Okay, we want to make our mind map to be more fun. So maybe we have like less words, more pictures, or maybe animation.
Yeah, sure.
Again?
How we are going to do our mind map?
There's an online website that the lecturer posted. I think it uses,
Popular.
yeah, whatever that is. But we can use that maybe, and then we share just the link. We share the link, and then we can just work it together. Maybe we'll just do the base, which was we put the name of the microorganism in the middle, and then each person will do a branch where they think is appropriate. But we on the WhatsApp group, I think we just discuss each person what they want to do, and from there we branch out.
Okay.
So we need to do more reading because us we've all seen each other's information. It's more of like fingertips of the whole entire microorganism. So maybe we can just do a more thorough research. And then with our other group members who could not join us today, they're joining us on WhatsApp, but they're not with us right now on the video call. So then from there, we start our work on the mind map. Because our mind map is due, hold on the 17th of October. So 2 weeks from now, approximately, if not less. I think we're capable of doing that, yeah. So using the app or the website that was given to us, we can add videos, GIFs or GIFs. How do you pronounce it? It's like moving pictures, but it's not a video.
GIFs.
Yes, GIFs. We can add maybe animation. So let's be creative and help each other out. So if you see one of us who did a branch, for example, and you think you can make it better by maybe editing a little bit or changing something about it, or maybe connecting it to a different point, I think we could learn from each other. We are, 3 or 4 universities, so I think I think we can figure something out. So anybody else who wants to add anything?
So that means we have to do our part individually and then combine it, right, in one booklet.
I want to see if we can I'll open one, and if you can share the link. So we share the link between all of us. So all of us work on one page.
Okay.
Yeah.
So who wanna go try doing the first one?
I think I'll try and do it by tomorrow.
And then you share the link in WhatsApp group.
Oh yeah, of course. Everything will be on WhatsApp. Like right now, after this, we'll have a short discussion on WhatsApp again to make sure that everybody gets what their which subtopic they want to talk about and what they want to discuss and talk and write about it. So each one will be as creative as you can, and then we help each other out through the mind map. So for example, I put an information which is wrong, or maybe I put information that should belong to a different group, like for someone else's branch. So you just edit it yourself and put it somewhere else. We're here to learn from each other. So that way it will help us to all learn from our mistakes, maybe from the other person's mistakes. At the same time, we correct each other. It's group work.
Okay.
Group work.
The informations we need to do in point form because I'm sure there will be lots of branching. So do we need to do in point form? No need, right? So we just insert the links and then a bit of branches.
Well, it's you and your creativity, but it's a mind map, so we don't want a lot of words. Like maybe make it like it's a mind map, so it has to be like, for example, keywords.
Make it short.
Yes. So keywords, like for example here, we put, for example, one branch staining. So you put from staining, you put another branch gram staining. From there you put positive bacillus. Then from the staining itself, another branch, maybe let's say acid fast. The result. Another type of staining result. So it's just 1-2 words. It's simple, not too crowded. At the same time, you know what's going on. If something needs a lot of explanation, maybe find an animation or maybe find a video or a picture which has full details. Like for example, microscopic observation. So maybe just put a picture labeled. If there's no labeled picture, get the picture and just label it yourself.
Okay.
Okay. We have to be creative as much as possible, and not very boring. Like something that you will study from.
Yeah.
Yeah. So something sweet, easy, and simple.
Yeah.
Sure.
Okay.
Okay. So all right. So yeah, Katrina joined us right now, and we're ending the meeting.
maybe Kat can just introduce herself.
Oh yeah, Katrina, can you introduce yourself with your name, student ID, and group? Katrina. Okay, I think the connection. She's at UQM, right?
I know Kat is UM2 as well.
UM2.
Yeah.
Okay, Katrina. Oh, okay, I think the connection keeps going on and off.
Yeah, it's okay.
Right. Okay, so Fatin, you're recording this. So if there's a way where we can share this recording with our 2 other group members so they know what we discussed over here. And then we need to post,
Yeah.
and we need to post this. But where can we post this? We have to post this at Cello
On YouTube.
on YouTube. Oh, okay.
I'm trying to figure it out and ask my friends. But I'm gonna upload it on YouTube.
Oh, okay. Oh, God. Okay. All right. Oh, wait. Let's check. Katrina, can you hear us?
Connection problem.
Katrina. Okay. I don't think she can hear us. Okay. Okay, Katrina, can you please state your name, your student number, and group? We're recording this video right now.
Okay. Hi, my name is Katrina Anaguan. Can you hear me?
Yes.
Yes, yes.
Okay. Hi, my name is Katrina Anaguan and I'm from University of Malaya and my matrix number is SEQ 150005.
Okay. Can you, we just had, this is a recap. We just discussed about the Clostridium perfringens, some basics about the toxins, how it looks like, microscopy, and identification. Is there anything you want to add to this Clostridium?
Me?
Yeah, Katrina, yeah.
It is gram-positive bacteria.
Katrina, your voice is breaking.
Family
Katrina,
and Arabic. Yeah?
your voice is cutting off.
Oh no. Okay.
So just give us like a short summary so that the connection stays as quick as possible before it disconnects.
It is non-motile bacteria, and anaerobic. It is gram-positive bacteria. It is a spore-forming bacteria encapsulated in tissue smears.
Okay.
So its vegetative cells are rod-shaped, pleomorphic, and occur in hairs or short chains. It has 5 toxigenic types, A, B, C, D, E, of which A to C strains are pathogenic for humans. So these colonies with double zone of hemolysis are produced when cultured at 37 degrees Celsius on blood agar overnight.
The blood agar has to be anaerobic. We have to
Yeah,
remember
and
that.
aerobic.
Yep. Okay. So, okay, great. So that's the same thing we've all, each person have gathered. So I think we need to do further research. And as we discussed on our WhatsApp group, each person has a specific branch to talk about. And then we will open a link for our mind map. And each person will work on that branch and then help the others. If you see another branch, for example, either lacking information or they have wrong information or their information can be placed in a better situation, we fix it and help each other out. Each of us learns from each other. Okay, and we will further do the discussions on WhatsApp later on. Okay?
Okay.
Okay, thanks all for meeting, and thank you, Fatin, for hosting our talk today.
Thank you.
Thank
Okay,
you.
okay, thank you. Okay, bye-bye.
Bye.
Bye, guys.
Thanks.
Fatin?
Okay, bye.
Bye, okay.
Bye.
Turn it off. Okay,
Yeah. Okay. Bye.
okay, bye.